Is that being recorded? Morning, everyone. My name is Chen Xie. Welcome to IOB seminar. Today I have the honor to introduce Dr. Langmead. Yesterday I learned that he actually had a Mandarin name, Shenben Tang. Anyway, if you ever used any sequence alignment tool, or if you heard of Bowtie or attended our IOB algorithm club, you recognize him as the creator of Bowtie 2, and also the author of the helpful lecture note on the trees data structures. Dr. Ben Langmead has been at the forefront of computational genomics for over a decade. The tool he created garnered over 77,000 citations, and it is still growing. His recent work on genome compression earned him the Best Paper Award in Recon 2024. And today he's going to talk about the critical problem of reference bias and its solution. Please join me in giving Dr. Ben Langmead a warm welcome.
Thank you, Chen. And I'm very impressed that you had that committed to memory. I once introduced a faculty guest for a seminar series and had it memorized. And my colleagues came up afterwards and said, "How did you do that? It's amazing." So it's a small thing that you can do to impress people. Okay, so welcome. Thank you very much. This is a fantastic audience. I'm really glad to be speaking to you about this. And I'm really talking today about a series of advances that have been since the early days of genomics. So, I'm speaking to a group of people who are going to receive their training in this new pangenome era. And so really what I want to try to instill in you is the tools are coming that are going to make the pangenome era a more successful era than the previous one. But you don't necessarily see lots of good information about this because so many of us were raised in the previous era, the single reference genome. So, when people like me were being trained, we learned quickly that there were these things called reference genomes. You've probably interacted with reference genomes. Many people here don't necessarily work on human, but let's just use human as an example. So if you've worked with human, you had to go download something with a weird name like HG38 or GRCH37 or T2TCHM13 or something like that and work with it. And you know that what you're downloading is essentially a string, right? You're downloading a file with a bunch of strings in it. The strings are the chromosomes and they have ACs, Gs, and Ts, and it's a huge file. And in fact, in a kind of comical demonstration of how long that string is for human, there was an exhibit in a museum in London where they actually printed out these bound volumes of the human chromosomes, the draft human reference genome. I hear it's not there anymore, so don't try to go visit it. Since then, right, so this was true when I was being trained. Since then, there have been, of course, many sequencing projects, short read sequencing projects, long read sequencing projects, and those projects have added a huge amount of genetic variation to our public catalogs. So shortly after I was a grad student, there was the 1,000 Genomes Project. That was a perfect early example. Uncovered tens of millions of polymorphic sites in the human genome. And in the long read era, we've had additional projects, perfect examples being the T2T human genome assembly, so telomere to telomere, perfect without any gaps, which is to say there were actually 5 gaps that they had to fill with magic. And then the Human Pangenome Reference Consortium, which is ongoing, and which has produced in its most recent release, the version 2 release, 466 human haplotypes, right? So if you picture that bookcase with one human reference genome, you have to multiply that by almost 500 to get the current kind of best high quality long read assembled collection of haplotypes we have for human. I put the wrong logo there, I'm sorry. HGSVC is a separate effort. It's also very good, but it's only sequenced on the order of dozens of haplotypes. I should have put the HPRC logo here by mistake. So we have all this wonderful variation. And let me go back. One thing that we might say is it would be a shame if we didn't somehow use this in our analysis of each new human that arrives, or You don't have to think of human, but let's just continue with the human analogy. Another way to say it is, if we don't use this, we're in trouble. Like we make mistakes. And so that's the reference bias problem. So let me just spend a few slides filling in some details on what we tend to call reference bias. Okay, so when we take a reference genome, let's say a single reference genome, and we align sequencing reads to it, sequencing reads are from another individual, right? A genetically distinct individual from the reference genome. So if the individual we're studying has patterns of reference and alt alleles, so alleles that match the reference and alleles that don't, and we're trying to align reads to it, we are more successful in finding the correct alignments for reads that have the reference alleles. Those match well. They yield long, unambiguous, nice exact matches, versus reads that have alt alleles, have non-reference alleles. Those sort of fail to match, right? And alignment tools sometimes are tolerant of those mismatches and gaps, hopefully, but all these tools have heuristics, and depending on the pattern of where those alt alleles fall, you can simply get the wrong answer when aligning those reads, or fail to align it at all. And so this can become a confounder. For example, as far as I know, the very first coining of reference bias in this area was back in the early days of RNA-seq with next generation sequencing. People noticed the following. If they were trying to study something like allele-specific expression from a gene in, let's say, human or a diploid individual, you've got the two haplotypes. Those two haplotypes, of course, have different variants on them. If one of them happens to have a higher burden of non-reference alleles compared to the other, it's a little harder for the aligner to find the correct alignments for those. Maybe in this example, in what I've called gene 1, maybe it's not too big a deal because there's only one alt allele on the maternal copy. So you might be like, okay, well, the aligner hopefully is smart enough to figure that out and not fail to align those reads. But what if there's a dense cluster of alt alleles on one of the two haplotypes? Now the aligner, again, the aligner uses heuristics. This could be enough to just completely defeat the heuristics of the aligner, and it might fail to align reads to the, in this case, paternal copy of that gene. But then again, what you were trying to study was allele-specific expression. You wanted to know the true biological question, the answer to the question, is one of the two copies more highly expressed? Well, this is perfectly confounded with that, right? So if you're trying to study that, reference bias directly gets in your way. It's a very serious systematic bias that you'll encounter. This is that paper, it's from 2009. So it's been 16 or so years that we've had this term reference bias floating around. Another example would be hypervariable regions, sticking with human. This is the MHC region. And if you align one data set to the reference genome and look at the MHC region, you'll see a very sad pileup with lots of peaks and troughs and those troughs are because the hypervariable portions meant that your reads just didn't have good matches, right? Your reads there's nothing wrong with your reads. Your reads are really from here, but the reference is so different from the donor that you just couldn't get good coverage. Here's another example. I like to show this because I feel like it's a particular, I'm not trying to give you nightmares, but it's like a particularly nasty example of this. This is a bisulfite sequencing experiment. This was bisulfite sequencing of several mice of two different strains. And with bisulfite, you're looking for methylation differences. So this is a differential methylation analysis. In this example, if you choose the reference genome to match one strain, you see one differentially methylated region. If you choose the reference to match the other strain, you see the opposite. And the truth is, neither are correct. The truth is there's no differential methylation. But depending on which of the two reference genomes you use, you're confusing genetic TPGs with what look like unmethylated cytosines and getting a spurious signal, and the direction changes depending on which you use. So like total nightmare. Not only is it spurious, but you get to pick the direction of the spuriousness according to the reference you choose. I can make similar points about long reads. Long reads help a lot with reference bias, but there's still reference bias because there are now large scale differences in references that are still perfectly capable of defeating long read aligners. So there's a separate set of issues for long reads as well. Okay, so all of what I kind of want to present today is all about working toward a world where we are not defeated by reference bias. In other words, if we go to design a sequencing-based diagnostic or therapeutic or something like that, that's going to work with humans, it really works with humans, not people who look like the reference genome, right? Related to that, and this is mostly going to be a talk where I focus on pangenomes that are for given species. But you could also look across species and say, well, there's another sort of pangenome problem brewing here, which is that in the long read era, we're getting all these new assemblies of different species across the tree of life. And we need to be able to wrangle all of those and use them effectively. And that's also computationally getting to be more difficult, right? So we're trying to reach a world where I like to say more is better. We want to reach a world where the increased availability of high quality assemblies is actually helping. And at the last minute, I swapped in the RefSeq plant plot I usually show there because I know there's many people interested in plants here. But over 14,000 distinct species available in RefSeq and growing. So I want to talk about two tools. One of them is pangenomes. I'm going to talk mostly about, so what is a pangenome? I'm going to talk mostly about a version of a pangenome where my definition is really simple. It's just a collection of genomes, right? It's just a bunch of genomes. If the genome is linear, then these are just a bunch of linear genomes. I'm not trying to cram them into any reference frame. I'm not trying to cram them into a graph. I'm not trying to cram them into a multiple alignment. I'm just letting them be strings and putting them in the same index together. And I'm going to call that a pangenome. An alternative, which is maybe the one we often hear about more often, is to put all the strings together in a graph. And you'll have a graph pangenome, where the paths through the graph correspond to the haplogroups that make up the pangenome. I won't talk about that so much, but I'll talk about that a little at the end. I'm going to argue that I very much favor the first path. I favor the path of keeping the linear references separate and linear in our pangenome representation. The second tool is, and this is the medicine you're going to have to take today, you're going to have to listen to me talk about the Burrows-Wheeler transform, because that's the fundamental computer science mathematical idea that's enabling everything that we're doing in pangenomics. And the computer science theory community has given us two wonderful new abstractions. One of them called the R-index, which I will discuss today. One of them called Wheeler graphs, which I'll only slightly discuss today. But both of them are two different versions of ways of taking the Burrows-Wheeler indexes that we all already use in tools like Bowtie and BWA, but making them useful for pangenomes by making sure that they don't grow as quickly as the pangenome grows. They grow much more slowly than the pangenome grows. Either you can do that for linear references, that's the R-index, or you can do it for graph pangenomes, that's the Wheeler graph. Okay, these are not my papers. I'm pointing to two papers from the computer science literature that are the ones that really have opened these doors. So from here on, I'm going to first of all say, why do we care about the Burrows-Wheeler transform? Why does it make sense that this is the computational basis for all we're doing with pangenomes? Then I'll talk about what are the compressed BWT indexes we have available today, what they can do in the third part, and then maybe what they can do in the future in the final part. Okay. What is the Burrows-Wheeler transform? All right, so let's say you're starting from your genome, or maybe pangenome, right? I only have one small string here. Just imagine, T could be anything. T could be 10 human genomes concatenated together or something like that. So you're starting with a string, which represents a genome or maybe a pangenome. The Burrows-Wheeler transform is just a way of reordering the letters of that string. So it's just taking the letters that you already have, putting them in a new order. Why would you want to do this? Isn't that weird? Like, they're in the original order for a reason, right? Why would we move them into some new order? Specifically, it puts them in order according to the alphabetical order of their right contexts. So if you take a letter and you look at what comes to the right, and just think about the alphabetical rank of that, that's what determines the new order that you put the letters in. Using order to accomplish different orders, to accomplish different goals is hopefully not too surprising, right? If you have a deck of cards and you want to play a game of poker and they start out looking like this, you should probably shuffle them, right? Because that would be boring. That would be a boring game of poker. Everyone would have a straight flush. So if you want to play poker, you should shuffle the cards, right? If you don't want to play poker, like maybe you just counted and it turns out there's only 51 cards and your kid has one of them under their bed or something, and you need to figure out which card is missing, it might be a good idea to first put them in order because then you can just scan through them and it's easy to find a missing card. I could use many analogies here, right? Like the index of a book, right? Which is kind of the analog that causes us to call these things indexes, is in alphabetical order. That's why it's easy to look things up. So order making it easy to use a data structure is not so strange an idea. But the particular order that I just mentioned might seem like a strange order, except I'll show you why it's not so strange. This is the more typical picture you will have seen. I think I call this the operational definition of the Burroughs-Wheeler transform because it's one way to build it, but it's not necessarily the right way to build it. And it's not even necessarily the right way to think about it, but you could think about it this way. It's as though we took our input, we took all the different ways of rotating it. Rotate just means take a letter off one end and stick it on the other end. All possible ways of rotating it, put them in order, take the last column. That's another way of obtaining the Burroughs-Wheeler transform. It's equivalent to what I said before. It's just another way to think of it. The Burroughs-Wheeler transform is at the core of tools that you may have used or at least heard of, like Bowtie and BWA. If you were to go to a big machine room in a big genome sequencing center and go up to one of the computers and ask it, what are you doing right now? Like, what are the computations you're running right now? Chances are they're querying an FM index. That's just one of the core computations we're doing constantly, genomics. The FM index is more than just the Burroughs-Wheeler transform. If you would like to know what else is involved in the FM index, this is the first of many times I'm going to say, you can check out my YouTube channel, right? Because I have a bunch of YouTube videos, including on the specifics of what do you need in addition to the Burroughs-Wheeler transform in order to turn this into an index. But the Burroughs-Wheeler transform is the main thing. The index is really just the Burroughs-Wheeler transform, plus a few other kind of small things. Okay. When you use the Burroughs-Wheeler transform, you can use it to solve pattern matching problems. So you can ask questions like, here's my query string. Is it present in the genome? Is it present in the pangenome? And that process proceeds in a series of steps. The steps tend to match longer and longer suffixes of your pattern until eventually you get to the point where it has identified all the rows of the matrix that begin with your query. Right? So if your query was ABA, you would run this process 3 steps. You would get to the point where now you know there's 2 occurrences of ABA in the genome or in the pangenome. You usually then have a secondary question is, well, where are they? Right? Okay, you could say they're right there. But you really want to know where they are in the context of the original string, right? Not in the context of this matrix. So you have another kind of query, which is called a locate query. The details are not that important, but there's 2 queries. You can do both of them quickly. If you can't do both of them quickly, you can't usually really use this in genomics, right? You really have to be able to do both these things and you have to be able to do them quickly. Okay, match queries, locate queries. So why is this order a useful order? Like, why put the letters in order by their right contexts? Well, interestingly, the first reason why people did this didn't have anything to do with indexing. It was not invented for indexing. They didn't have us in mind. It was invented for compression. And it turns out that compression and indexing are almost the exact same problem. It's hard to see at first, but when you study it, you realize they're really both about sucking all the redundancy out of the representation and then leaving enough breadcrumbs so you can get back to the beginning, to where you started. But originally it was invented just for compression. And we can actually see already with just a simple example why it might be useful for compression. If we apply the Burroughs-Wheeler transform to this string here, which I can't say out loud, right? Something bad will happen. To this string here, you can imagine there's 3 instances of E told juice in this string. And each of them are preceded by the same letter, which is B. So if you put everything in order according to the right context, then those 3 B's, which all have the same right context, should come together in the Burroughs-Wheeler transform. They'll be right next to each other in the Burroughs-Wheeler transform. And there they are. So if you look at the bottom there, the 3 B's came together. If your job was to compress the string, would you rather work with this or this? right? You'd much rather work with this thing on the bottom, right? That's going to be super easy to compress because look, all the redundancy is right there on the surface. It's easy to just, you know, you could replace the three B's with like a little sign that says B times 3, and that's maybe a good way to represent that stretch. So and you can go the other way. You can go back, right? So that's why it's useful for compression. You can rearrange the letters so that all the predictability is right there on the surface. You can then compress it. And then you can undo those two steps in reverse order and get back to the original. So that's something special about the Burroughs-Wheeler order, and it is essentially why it's useful for both indexing and compression. So now let me show you a pangenome. So here we go. It's not really a pangenome, it's just another silly example, but I use this example. This is 3 copies of Row, Row, Row Your Boat concatenated, right? So I wrote it out on 3 lines, but just imagine they're concatenated. And I like this example because there's repetitiveness both within a row, 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 row. And there's repetitiveness across, right? Because the 3 rows are identical. If we want to index this as a pangenome, we would like to be able to remove both kinds of redundancy. Like we want to remove the within row and the across row redundancy. So we think the Burroughs-Wheeler should help us. So we think if we apply the Burroughs-Wheeler transform, we should get those equal letter stretches, those runs that we saw in the Betelgeuse example. And we do, there they are, right? So if you look in there, there's some nice long runs. This is 9 W's in a row. This is 9 R's in a row. This is even better. This is 12 O's in a row. So man, we're doing such a good job here. And in fact, if we were to try to compress this, we would probably just write it down this way, where instead of writing 12 O's, we say O 12 times, right? We would represent it like this. And so we started with 64 letters. There's 64 letters in those 3 strings. But if you put it through the Burroughs-Wheeler transform, there are only 15 runs, which we can then represent. And so if you wanted to give an approximate compression ratio, we might take the number of letters divided by the number of runs, and that gives us 4.27. 4.27 is good. It's greater than 3. And one thing that indicates is that we must have taken advantage of both kinds of repetitiveness, right? Because if we were only taking advantage of row, 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 oh, sorry, that was ambiguous. If we were only taking advantage of the 3 rows being identical, we'd get 3 as the compression ratio. But we're also taking advantage of the word being repetitive, the word row. And so we're getting something better than 3. So this same idea that makes the Burroughs-Wheeler transform useful for indexing things like a single human genome would seem to also make it appropriate for indexing many human genomes or something like that. Okay, that wasn't really a pangenome. So now let me show you a real pangenome. Okay, that's still not really a pangenome. But of course, a real pangenome, they're not identical, right? Like you're not putting identical sequences in there. There's no point. You're putting sequences that are like different humans' genomes, right? So you expect there to be some variation. And that variation is going to reduce our ability to compress the sequences relative to if they were identical copies, right? Because now the right contexts are a little bit less predictive of what's going to come to the left. Right? It's just a little bit less regular. So if we put this through the Burroughs-Wheeler transform, we get a new set of runs, you know, more, right? So we end up in this case with 21. We had 15 before. And so 64 divided by 21, that's just a little bit over 5. I'm sorry, over 3. So our compression ratio is now a little over 3. And it's worse, but it's still good. I do that still good, right? And 3. And hey, we've got 3 copies. So like we got pretty good compression. We got maybe as much as we could have hoped for. Okay, but this is a little bit more realistic compared to my previous example. Okay, enough with the toy examples. Let's go on to human. So what would it look like if we started with several human genomes, distinct human genomes, put them through the Burroughs-Wheeler transform, and then asked how many runs are there in the Burroughs-Wheeler transform? That's our kind of proxy for how much distinct information is there across all those genomes. And so we have now wonderful data sets for investigating this question. And this is one of them. This is the Human Pangenome Reference Consortium Release 1, okay, which has 94 human haplotypes in it. And so as we add more haplotypes, the amount of data that we're putting in is going up linearly with the number of haplotypes. And then if we look at the number of runs in the Burrows-Wheeler transform, it's going up sublinearly. It's going up more slowly. So you can picture that down here, right? Like, so this black line here, that tracks with the number in the green column. And so you can see as you add more genomes, you start to add, instead of 3 billion bases for each haplotype, closer to 100 to 200 million bases or not bases, but runs per haplotype. And this is the release 1 of HPRC. You might wonder, well, what about those 466? Well, we can do this with those too. And the final R that we end up with is 5.25, right? So we started all the way up here at 3.33. So when we multiply the number of genomes by almost 500, we multiply R by what? 1.6 or something like that. So this is fundamentally the reason why the Burrows-Wheeler transform is kind of a perfect tool for pangenomes, right? When you find the Burrows-Wheeler transform and you operate with the runs that come out, then you are really working with an index that's growing only with the amount of non-redundant sequencing. And you need that, right? Because otherwise you run out of space pretty quickly as you add more pangenomes. Okay. So fundamentally, as a tool, it seems like the Burrows-Wheeler transform kind of has what it takes, right? It can give us the compression that we need. It's not just for human. So I'm showing examples from other pangenomes that we work with. I was talking earlier today about multiple of these, like maize one and also the potato one. Right? Somebody was saying, which potato one did you do? And now you can see down there, it's reference number 6. In all cases, if you look at the compression ratio, the human is always, man, comes out way on top. Humans we're just so similar. But then you also got a very, very, very attractive compression ratio for all these pangenomes. Like, would you rather work with something that's the original size or 30 times smaller? So even for the quote unquote worst result here, it's still extremely good. Okay. So the Burrows-Wheeler transform as a tool is extremely attractive, but we need the corresponding algorithms. Like, we need to be able to build and use indexes. And what's really important is we need to be able to build and use the indexes so that they grow with R. And R is, again, the number of runs in the Burrows-Wheeler transform. We would much rather it grow with R than it grow with N, where N is just the number of bases in the input, right? So that would be linear. We don't want it to grow linearly. We want it to grow sublinearly. So we need growth like R. I'm using some big O notation on this slide. Don't be intimidated. This just means grows like N or grows like R, proportionally to N, proportionally to R. So the original work that Bowtie and BWA is based on, that is not compressed in this sense. It doesn't grow like R. It grows like N. If you index 10 genomes, it's 10 times bigger. Okay? Not long after that, though, the theoreticians were already kind of eyeing this goal of trying to make compressed indexes. And so a few years later, there was a proposal for how to do one of those two queries that we discussed earlier, the match query, in big O of R space instead of big O of N space. And they succeeded, but only for the match query, not for the locate query. So this was like part way to the promised land, but not quite there. And so, but look, that only took 5 years. So you might think that in 2010 or so, we got the locate query. But it took longer than that. I'll show you that in a moment. But anyway, the idea that got us to the big O of R on the match query is illustrated here. I'm not going to walk through this. That would take far too much time. However, this is another instance where I'm going to say, I have a YouTube video. And specifically, if you go to my Burrows-Wheeler indexing playlist, video number 19, is the one that you can watch to better understand this idea. But the point is, everything that you see on the screen here is something that either straightforwardly grows like R, or even if it looks like it grows like N, it actually can be represented in just R amount of space. Like here's a length N bit vector, but there's only R set bits, which means that we can represent it in R space. Okay. So that's the idea behind the first step. So again, what do you think happened? Maybe 5 years later, we got the locate query. No, it took another 13 years. So 13 years later, there was finally a publication that said, aha, we can do them both in big O of R space. And that was something called the R index. The R index is the foundation for everything else I'm going to talk about. This was a very important breakthrough. And just like the original FM index paper, it came out in, a super wonky egghead computer science journal or conference. This is the SODA conference, one of the main theory conferences. And the FM index came out in the Fox conference, one of the core computer science theory conferences. So it came from very much a place of core computer science, which is where things, I'm kind of like a middleman. I look for these sorts of things when they happen. I grab them and I say, oh, let's see if we can apply them. So the idea behind the big O of R locate query is also something I have a video on. This is quite involved. I think it took me at least a year after initially reading the paper before I finally understood how it does this. But anyway, it's all based on something called the total lemma, and I have a series of videos that explain this. Okay, but the point is, in just a small number of years ago, we finally had access to the theoretical data structures that could allow us to index pan-genomes in big O of R space, an amount of space that's proportional to the number of runs in the Brooks-Wheeler transform, compressed space. Okay. What we had to do next was apply it, and there was an immediate problem, which is that we couldn't build them, right? There was a description of what the data structure should be, but it wasn't possible to build it for human genomes because there just weren't algorithms good enough to do it. So step one was to come up with quick and dirty algorithms that were capable of building the R index for human genomes. So that was the first time I jumped in and started collaborating with this group. These are several authors of the initial papers using a technique called prefix-free parsing. Okay? And it is my goal to eventually have videos on this, but I don't. So you'll have to read the papers if you want to learn about prefix-free parsing. And we applied it to human genomes. And that prepares me for the next part of the talk, because now we're going to talk about what they can do. Right? So we have the R index. We have reached a place where we have data structures that grow like R. We need to be able to apply them. Can we? What can they do for us in bioinformatics? Can they do stuff today? So the first thing we showed, and a little setup. I'm going to talk about this progress in kind of like 3 stages in terms of how sophisticated are the things we can do with the R index. Stage 1 is going to be looking for matches, just straightforward. You give me a string, I looked and I try to tell you if it occurs in the reference and where. Straight up substring matching. That's a basic functionality that all full text indexes need. So I need to first demonstrate that we can do this. And then a little bit more sophisticated than that might be classification. If you give me a string and in my index I have several genomes from the tree of life, can I tell you which of those genomes the strings seem to originate from in a principled way? That's usually a more sophisticated problem, right? That's the kind of problem that tools like Kraken, for example, are solving. So can we solve those sorts of problems with compressed indexes? And then ultimately, we want to be able to do like read alignment. Like we want to be able to do the same things that Bowtie, BWA, Blast, and similar tools do, but in compressed space with respect to compressed indexes, pan-genome indexes. So I'm using a Pokemon analogy here that used to work really well with students. I think it's maybe aging a little bit, but since I'm talking about something called the R index, I'm going to refer to these as Armander, Armelian, and Armazillion. So our goal is to get the Arizard, right? If we get the Arizard, a little spoiler, we're not at Arizard. Still the goal. Okay, so we start with Armander. So the initial paper where we were able to build R indexes over human genomes demonstrated in this plot, which I always hate to show because it's such a plot, that we were eventually able to get to the point where once you have enough individuals in your pangenome, the big O of R representation is much smaller than what you would have got with an FM index. This was totally predictable, right? This is just purely based on the principle we already discussed. It would have been very surprising if this wasn't true. So this is really just a confirmatory result. The other thing that was a bit interesting is we were also eventually able to be faster than bowtie on a very specific thing, which is the locate query that I mentioned before. So once the pangenome grew to a certain size, there was a point where the R index became somewhat faster than bowtie. And, you know, we hope for it to be faster than bowtie at all points. But this was early days. We did not have a very tuned implementation. Bowtie's been very, very, very tuned, right? So we were just happy to have there be any point here where we were able to achieve faster speed than bowtie. So that was the initial confirmatory results that basically established we can do char mander. We can do RMAN. Okay. Okay, so now slowly transitioning from RMander to Armelian. There was a paper that was published by Max Rossi and others. Max Rossi now at Illumina, Marco Oliva now at NVIDIA. I hope he has stock options. Great for him. They studied this approach called MONI. MONI was a kind of fast algorithm that was able not only to find substrate matches, but basically to find the landscape of all possible maximal matches. Right? So this was something called matching statistics. You can also think of it as MEM finding, maximal exact match finding. If you can find all the matching statistics, or if you can find all the MEMs, then every other question you might ask about substrings is just a special case of what you've already found. So it's sort of the ultimate exact matching information. And once they did that, we grabbed onto it and we said, we would like to use that for classification. And there's a whole set of projects I could talk about. There's too many of them, so let me just highlight a few. These three, Spumoni, Movie, and CallBWT, which are all on the road to what I would say our biggest success so far is, which is that we can use compressed indexes to do classification, both multi-class and taxonomic classification, and we are competitive with what alignment-based methods can do. So I want to tell that story next, briefly. Few minutes left, so maybe I'll start to go a little bit quicker. Did we start at 11:30? Okay, great. Okay, short version is, because we are finding this landscape, look at the middle there where it says MONI, because we're finding this landscape of all possible maximal exact matches, you can start to think of those matches as kind of like test statistics, right? So if I want to know whether this read came from this genome or that genome, I really want to know, are my matches more significant when I match them to this genome versus that genome? So we implemented a way of computing not the matching statistics, but this kind of shadow of the matching statistics called the pseudo-matching lengths. For technical reasons, it's much more efficient to compute the pseudo-matching lengths. And we applied them in a framework that said, well, if the pseudo-matching lengths we find with respect to one category, like we're doing classification, one class, if the pseudo-matching lengths we find with respect to one class are significantly longer than a set of null matches, right? You can imagine matches to random sequence, or just take the same sequence you were matching and reverse it. Reverse it. That should have no relationship to anything in the database. How long are those matches? Those could be considered null statistics. So if you compare the lengths of the matches you get with null statistics, that gives you a mechanism for doing classification. If the test statistics are much longer, right, if they're shifted to the right, have a fat tail, you would say, yeah, that looks like classified. If they're on top of each other, you would say, this is indistinguishable from a random match. You would not classify it. And so we applied it in a setting where we were competing with Minimap2, which is kind of the gold standard for long reads. This was a long read setting. And we found we were super duper fast, which was great. We kind of knew we were going to be super duper fast compared to Minimap, because Minimap does a bunch of very sophisticated stuff, more sophisticated than what we're doing, but we were a bit less accurate. So we worked on it a bit harder, and in particular, in a second project, one of the advantages Minimap has over us is it doesn't just try to find those initial matches, it also tries to chain them together. So you may have studied chaining in some contexts. It's sort of like, well, it's not enough to just figure out this part of the read matches and that part of the read matches. You need to know if they are collinear with respect to the reference that you're aligning them to. And usually you would do this with chaining. And we came up with a kind of quick and dirty strategy that does something like chaining, but it does it all in the context of our compressed index, where we're able to find conserved regions of the pangenome, not by building a multiple sequence alignment, but just by looking for things called multimoms. So sequences that are conserved across all but unique within each. And then we can paint the index according to the multimoms that we find. And then we can paint our matches with respect to which multimom they fall into. And we can then draw on these two sources of information, both how long are the matches, right? These look like nice, these look like somewhat longer matches than these, right? So maybe we would say, yeah, these are significant. But furthermore, the coloring of those matches is consistent with the read having come from like here, right? This part of the multiple alignment, right? It's like half blue, half red, right? So it's giving you some collinearity information with respect to the reference. And this was kind of the final boost that we needed to finally be right up where Minimap 2 is in terms of classification accuracy, but still much faster. That's kind of our first big success. I'll just say this very quickly. After the R index, there were more theoreticians who developed a new, you could think of it as a new version of the R index that had different theoretical advantages. They proved different and improved bounds. But more importantly, they rearranged the data structure in a way that, computer scientists here, you'll know what I'm saying, had better locality of reference, right? So in other words, querying this data structure was friendly to the memory system of the computer, whereas the R index, it was highly unfriendly to the memory system, and you had to spend a lot of time waiting for data to be retrieved from memory. This move structure idea was radically in the direction of good locality of reference. It led to huge speed boosts relative to the R index based methods. And if you would like to learn more about that, I also have a video. Next slide. But that particular work then spurred others. We got contacted by Rob Knight's lab. Rob does a lot of microbiome analysis. And he said, we tried using Minimap to align reads to single reference genomes. It was not good enough for removing all the host reads from our data sets. We were trying to remove the host reads, the human reads. So we're looking for good pangenome tools. Can we use Movie? And so we collaborated with them on that, and they continue to use it. So here's a more recent same Movie-based pipeline. So here's a compressed index, a fast compressed index being used in practice by people who do real metagenomics. Here's the video. That's where I explain the move structure as well as my point about why it's fast. Okay, so with my remaining time, I want to say, what can we do now? Armander, Armelian, check. We can do both those things. Armelian is in fact really competitive, even with alignment-based approaches. So I'd say that's a very strong sign of success. Everything we do is not choosing a fixed value of K, right? You guys maybe have heard of K-mers, you maybe use K-mer-based approaches, you maybe thought, what should I set K to? And then been horrified that there's no answer to that question. You don't have to answer that question with any of these methods. There's no fixed K that you have to set. It has excellent speed thanks to the move structure, which is what reoriented things for better locality of reference, and it's a huge improvement over the FM index if you're doing genomes. But what can't it do? Well, we're still not to RSR. It's for a rather technical reason, and rather than go through these bullets, I'm going to skip ahead to, I'm actually going to skip all the way ahead, sorry, to a picture that I have later. I'm going to skip back to this in a moment here. So we do have work together with Christina Boucher and Max Rossi and others where we have implemented an R-index based aligner. It's very similar in its interface to BWA-MEM, for example. And what we find though is that it's not yet competitive on how its speed scales with pangenome size. And the main reason for that is that these are graph aligners. Graph aligners are aligning to a compressed representation of the pangenome. Ours is compressed, but not in the same way that a graph is compressed. A graph is compressed because it takes lots of homologous sequences and it makes it one thing in the representation. We compress in the sense that we get the big O of R style scaling, but we don't collapse the sequences that we're compressing, right? We just benefit automatically from the big O of R. Because of the collapsing into a graph, Those tools do many locate queries. They only have to do locate queries with respect to the nodes in the graph, which are pre-collapsed. We, on the other hand, have to do locate queries proportional to the number of haplotypes in the pangenome. That's why we're not doing so. So that's why we're not quite in R is R territory. So with my remaining couple of minutes, let me just quickly go back and say one more thing. Two more things. Everything I said about pangenomes should be taken with at least one grain of salt, which is that in the future, if you're going to study, for example, the screen's not in sync with my laptop anymore. Sorry.
Change.
I've moved around several times since this was on the screen, but now you can't see it. Okay, I'm going to paint a picture with words for you. So in another project, what we've proposed is that you don't necessarily want to use a pangenome in all scenarios. So let's say you have whole genome DNA sequencing from a new person. Another strategy would be take a little bit of those reads, put them through today's wonderful imputation panels. We have fantastic human genome imputation panels now. Impute a personalized diploid reference for that human, index that, use that. In other words, do everything again, but using the imputed personalized reference genome. Sounds like more work, but it's strictly better in the following sense. A pangenome has many genetic variants in it, and the individual you're studying has, hopefully, a subset of those variants. But only a subset, only a small subset. So the pangenome has a bunch of stuff that's not in the donor, which means that it's more ambiguous than if you were to have only the alleles that are present in the donor. And all signs point to this being the superior strategy if you can do it. Can't always do it. Sometimes you don't have DNA sequencing, you have ChIP-seq or you have RNA-seq, and it's not easy to do imputation. But if you can, all signs point to personalization being the way to go. Personalization, by the way, uses the Burroughs-Wheeler transform in nearly every step. Because if you crack open these tools, even the imputation tools use the positional Burroughs-Wheeler transform, for example. Burroughs-Wheeler is the fountainhead for pretty much everything you see here. All right, allow me to skip all the way to this slide. Okay, so my final message is, for pangenome indexes, we'll know that we're in the right world, that we're in a good timeline. If as more and more long-read assemblies appear and we put them into our pangenome references, our answers get better. We are not currently there, and I think the primary reason is because we are too lossy at the moment in terms of what happens when we put the genomes into the pangenome, all right? So for example, one of the things that we make a mistake about when we make graphs is that we don't remember what the original haplotypes were. And so there's all sorts of paths through the graph that represent fantasy recombinations between these haplotypes, not real things. That's a ton of extra information that's not real or helpful and can only distract us from finding the real alignments. Hamerization is the other major thing that we do that's incredibly lossy. We put all this money into making beautiful T-to-T long-read assemblies, and then when we go to index them, we shred them into the tiniest pieces, even smaller than short reads. What's wrong with us? We shouldn't be doing that. So full text indexes are the solution to that problem too. And so I hope I've convinced you that of the problems that remain for making pangenome indexes practical, many of those problems are only technical, and we can solve them. And the computer science literature is giving us good methods for solving them. I mentioned several times my playlist. This is just the slide where I have the playlist here. I'm also happy to send it to you, shoot me an email. And I want to thank everyone who worked on this, and especially our funders, NIH, NSF, and an NSERC Canadian grant to my PhD student, Nate Brown. And thank you. All right, I used 50 minutes.
That is perfectly timed. We have 10 minutes of QA.
So I'm actually curious. So I do a lot of deep learning stuff, and I try to apply whatever I see in a deep learning conference to biology. And I'm actually curious, what made you first want to go into looking at let's say pure mathematic conferences, papers, and all that stuff, and how do you stay up to date? Because I know
Yeah.
computer science moves really, really fast.
Yeah.
So like,
Yeah, I mean, it was purely out of necessity. When I first started grad school, it was the dawn of the second generation sequencing era. It was 2007. Solexa, which is now Illumina, was brand new, or it's been absorbed by Illumina. I was sort of tasked with figuring out, well, how do we analyze these data sets? At that point, if you wanted the index, first of all, if you wanted to use an index, and it was pretty clear you should, right? Like there was actually some debate at that point as to whether to use an index or not. And there was one very early tool called MAQ, which did not use an index. And I think we were temporarily fooled into thinking we didn't need an index, but that changed as soon as the data sets got bigger. If we needed an index and we needed it to be practical, that meant that the index had to fit in memory and to fit in RAM. This was at a time when 2 gigabytes was like a decent amount of RAM. So like the machine on my desk had 2 gigabytes. So I was like, okay, it has to fit in 2 gigabytes. There were very few actual options. You know, the standard options like suffix trees, suffix arrays, far too big. So it really was out of necessity. You had to go to the computer science literature where people were studying, you know, the most compressed representations that were available then. There was just no choice. This idea had been around for already 7 or so years, which I don't know, does that make it old or new? I'm not quite sure. In computer science theory, there are often long gaps before things actually get used. And so it was just out of pure necessity. And I knew which part of the literature to look at because it was very clearly a text indexing problem, right? That's the name of an entire subfield of computer science. Now, stubbornly, I continue to just look at the computer science literature usually for inspiration for where to go next. That isn't necessarily the right strategy, but it's my strategy. But it continues to be the case that if you have a pretty good view of what are the top 10, top 15 problems that people have to solve computationally in bioinformatics workflows, and then you monitor the computer science literature, you'll find more connections than just what I talked about here. Like I'm also very interested in sketching, for example. The computer science literature, sketching and streaming algorithms was hot. I'm not sure it's hot right now, but it was hot for a time. And those moved over very nicely to genomics as well. So I don't know if it's the right general strategy, but if you are trained in a particular area and you can read literature in that area, but you also know the genomics problems that are out there, sometimes you can make one of these very win-win connections.
That one of your work, like recent work, was with the metagenome, where you have multiple genomes. So I was thinking, like, whenever we try to do phylogeny, like not in a pangenome sense, but across organism sense, then we are usually looking at changes or like a single nucleotide changes, but not usually at like variation or probably structural variation or copy number variation. Is it possible to like translate this approach on pangenome to like whole genomes from multiple organisms?
Oh, certainly. Yeah, I mean, like, so that my argument about N over R was about the compression ratio. I only showed you examples where it was many same species genomes. But you can sort of mentally simulate what happens if you had multiple collections and now you put them all into one index, which is that the N's are going to add and the R's are probably mostly going to add. And the reason I say that is because two different clades in the tree usually don't have very much similarity to each other. So you don't usually get the runs getting longer. You usually just get the runs interleaving. And so R is just additive in that case. So to a first approximation, in some sense, you get to keep all the compression that you got in each of the clades, but there's probably no additional compression that comes from combining the clades, or it's rare to get much additional compression. But that said, it's perfectly possible to do. And so the indexes that we use for these multi-class and metagenomic problems, we do this. We build the pangenome out of many genomes for species A, many genomes for species B. We have to keep track of which genome is which clade. So we need to have a way of saying, in the computer science literature, this is called document. We call them document identification, right? So you have to be able to distinguish which document your matches fell into, which is to say which genome or which class. And multiple of the papers that I discussed are mostly about how do you do that. how do you keep the document information linked into the rest of the index so that you can answer those questions? So, and there's even specialized versions of those structures that are in particular for taxonomic classification, where you need to be able to go up the tree and find lowest common ancestors. So all those things are very possible. It's too much to hope, and indeed it doesn't happen, that when you combine different species, you don't get more compression than you got inside the individual species. You mainly just get an interleaving of the N and the R, and they just add together, you know, the N in the numerator and the R in the denominator just basically adds.
Okay, so I have a question about, in the beginning of your talk, you mentioned that you prefer sticking with like a linear model of genomes as opposed to shifting totally to like graphs.
Yeah.
So I was wondering if you could talk more about that and why you prefer the linear.
Yeah, I skipped over my sort of soapbox slide about that earlier. I talked about the two forms of lossiness, right? About if we forget about which alleles co-occurred on the same haplotypes and we allow these sort of fantasy recombinations, then that's loss, and that can reduce the quality of our alignments. And then the k-merization. So one reality of how the graph aligners that actually exist today are implemented is that they're k-mer based. So the underlying index is k-merized first. At least the initial steps, the steps that look for seed hits, use k-mer indexes. But my other complaint, this is a separate complaint about the graph-based methods, is they usually start with multiple alignment, right? And that seems like a good idea, except for multiple aligners are not actually capable, really, of aligning human genomes. You have to do surgery on them first. You have to take out the difficult stuff. You have to get rid of the difficult repeats, get rid of some of the difficult large-scale structural variation, because multiple aligners are not well suited to deal with all that. And so the pangenome graphs that are published and used often have, they sometimes go by the name of burn regions. It's like regions that they took out because it was too complicated for the multiple alignment. So that's the other reason why I certainly don't prefer to start from a multiple alignment based graph, because I just don't think you can get those without doing something, you know, violent to the sequence. So if we keep them as strings, there's still notions of it. There's another slide I skipped over quickly. There's still notions of a graph that we can extract from that collection of strings. It's not based on multiple alignment. It's purely based on similar motifs in a graph, right? We find motifs in a graph that are collapsible. They tend to correspond to conserved sets of columns from the multiple alignment, but they don't have to be. They could be any conserved motifs in the graph. And there's a technique for doing this where if you start from the strings and you collapse and you collapse and you collapse and you collapse using this method, you're guaranteed at every step to have what's called a Wheeler graph, which is a special class of graph that is easy to index. Right? Not all graphs are easy to index. This is why they k-merize, is because the graph they begin with, it's very hard to build an index, so they k-merize in order to make the index possible to build. But this is another strategy that ensures that you can build an index, and it does not use multiple alignment. So I would say if your graph looks like this, and there's good reasons to do it this way, it may even solve the problem that I highlighted on this slide. If you make this kind of graph that I'm talking about, then it doesn't have to be based on a multiple alignment, which means you get to keep everything. And I just think I think it's important to keep everything. And I feel like this should be uncontroversial when you consider how much money we spend to make the T2T genomes, right? Like that's incredibly expensive. We should keep everything. So that's my opinion. And so when you start from multiple alignment, I don't think we're good enough at multiple alignment yet. Or it may even be that we have to give up on the idea that there even are common coordinates such that we can do multiple alignment. We're not good enough at that yet in order to keep all the data at hand.
Does someone have the last question? That's about time. Great, thank you.